Dear ladies and gentlemen, dear contestants, innovators, change agents, as I actually like to call you, distinguished members of the jury, as well as a lot of science aficionados that are either here with us in present or also watching us online via our live stream, welcome to the third edition of the Falling Walls Lab Marie Skłodowska-Curie competition. A big round of applause. My name is Raluca Angel, and I will be your moderator or your host for today. I promise I won't crack too many jokes. I only have a couple of chemistry jokes, but I'm afraid they won't get a good reaction. But one thing that I can tell you is that this is a fantastic opportunity for us all to actually discover how important research, innovation, and science is to our daily lives. It is of paramount importance because actually, all these researchers and scientists can actually come up with, let's say, important solutions to both today's and tomorrow's extremely pressing challenges. Well, we're all extremely lucky to learn from 30 researchers, 30 Marie Skłodowska-Curie Actions ongoing fellows that are going to pitch for 3 minutes their research scientific projects. We're going to learn and actually discover a lot of breakthrough ideas, as well as a lot of new ways of solving some of the issues we might all face. What's going to happen is that for the next 2 hours, we're going to be together, learn a lot, as well as end up analyzing a lot of these research projects. The winner of today's competition is going to go to Berlin for the grand finale of the Falling Walls Lab that is going to take place on the 8th of November. Let's say the winner is going to have even a further difficult task, and that is to compete with 100 other researchers from around the world for the fantastic title as Falling Wall Young Innovator of the Year. What I can say is that all the 30 people that you're going to see here on stage, they're already winners. They're winners because of the fantastic task that they have taken in their lives to actually change our world through their research projects. But they have also been selected by the Marie Skłodowska-Curie Actions from 200 other researchers. So you will actually see the crème de la crème of the MSCA. I don't want to take you up for much longer. I would like to only tell you that we have distinguished members of the jury that are going to analyze from their perspective and experiences all the scientific projects that are going to be presented here in front of you today. They will look at 3 very important elements. One is the innovation, breakthrough element of your presentations. Second is the relevance and impact they have on society. And third, an extremely important one, is actually the structure and performance of your presentations. For all the 30 ongoing MSCA fellows, you know this is part of a very important task that the Marie Skłodowska-Curie Actions have, and that is to actually equip you with the best skills you can get and the international experiences so that you can have a successful career next to the funding you'll receive for your research projects. So I would say we're all here with you. This is a fantastic time for you to learn. Too bad we're putting you on the spot for that. But we're looking forward to seeing how you perform. I would like to invite here in front Jean-Philippe Gamel. He's member of Commissioner Tibor Navracic, European Commissioner for Education, Youth, Culture and Sport, who is the chair of the jury and who will address a couple of words on behalf of the jury. Jean-Philippe, please, you have the floor.
Many thanks, Raluca. First of all, I would also like to welcome you all on behalf of my Commissioner Tibor Navracic. As Raluca mentioned, he's Commissioner in charge of education, culture, youth and sport. He's also responsible for the Marie Skłodowska-Curie Actions. This year we already have the 3rd edition of the Falling Walls Lab event dedicated specifically to Marie Skłodowska-Curie Actions. But the big novelty is that for the first time we are part of the bigger Research and Innovation Days, and this should help us to boost the visibility of our event. Over the last 3 days, the Canal has witnessed many heated discussions on the future of research in Europe. And that's great, because one of the main aims of the Research Innovation Days are precisely to provide a space for policymakers and for stakeholders to discuss the implementation of the Future Horizon Europe. But the other big aim of the Research and Innovation Days is actually to showcase some of the research projects which the EU is already funding and to demonstrate how science can actually help us to improve our lives. And that's precisely what our young scientists are going to do over the next couple of hours. As Raluca mentioned, all the 30 participants are MSCA fellows, and that's very important. I won't have time to say much about the MSCA. I would simply like to highlight that this program is the main EU's program supporting top-level researchers, either while they actually are doing their PhDs or just after. The program enables them to do their research abroad, and by this, what we try to do is first, to help them to have a true European experience. Second, to develop new skills which will be useful for them. And third, to boost the employment prospects. The Commission is also very committed to make science and research much more easily accessible and understandable for everybody, including young people, in order basically to inspire them to become scientists themselves. This is why every year we organize the Science is Wonderful exhibition, taking place right now just next door in Touré Taxi. If you have not been yet, please go. That's why we organize every year the Researchers' Night, tomorrow. Again, make full use of it. And that's why we organize this Falling Walls Lab, specifically dedicated to MSCA. I would like to introduce the other members of the jury, and I think there should be a slide here behind me. So first, in the jury, we have Timo Hapeletto from the Ministry of Economic Affairs and Employment in Finland. Timo, maybe you can just wave your hand so that we see who you are. Then we have Mostafa Munir Shobro, who is the Vice Chair of the Marie Curie Alumni Association. Mostafa. We have Michela Poudou, who is the CEO of Helixa, and who is also the winner of the EU Prize for Women Innovators. We have Guillaume Corradino, who is the director of the Pint of Science Festival in Belgium. And very soon we will have also Wiebke Rössig, who should join us in a couple of minutes, and who is from the Museum of Natural Sciences in Berlin. Communicating science is quite difficult. Doing so in 3 minutes is almost an art. That's what our young scientists are going to do right now. They have worked very hard to prepare their presentations. They have rehearsed a lot over the last couple of days. I won't keep them waiting any longer. Good luck to all of you, and let the competition start.
Thank you very much, Jean-Philippe. And the only thing between you and actually getting to speak to the first researcher is just me explaining the rules. We have a lot of empowerment through the number of 3. That is, we have 30 competitors, 30 researchers, and all they have is 3 minutes to pitch their research program. But within those 3 minutes, after 2 minutes and 30 seconds, you will see their slides actually change, and the slide with question will appear, which means that's the time for you to take the floor. Should anybody have any question, feel free to address it, but do keep in mind you need to both address your question as well as get an answer in 30 seconds. So it's a challenge both for you as well as for the researcher that will try to answer your question. We've all been there. We've all tried to address questions that would take 1.5 minutes. Please don't do it now, because then you won't even have the time to hear what the scientist actually has to say. So 3 minutes, but 2 minutes 30 plus questions, after which the timer will stop and you will see me becoming incredibly unpopular with the 30 researchers, because I will need to come here and put an end to their presentations. Another element that is quite important is for you to know that you also have an active role. We will also have, next to the 3 key awards for the 3 top winners of today's pitching discussions, let's say, we will also have an audience award. So you will all receive a ballot with votes that we would kindly ask you after the 2 batches of presentations to just give it at the back to our colleagues. And then according to your popularity vote, we will also have your 4th award, which is the audience award. Remember, 3 conditions: element of breakthrough, element of impact on society, as well as the structure and performance of the organizations. We're live streamed, so please watch out what you do, be responsible. But we're also on Twitter, so should you want to actually share on Twitter, please make sure that you use hashtag RIDaysEU as well as hashtag MSEA. And one last thing, as you also hear at the movies, please make sure all your phones are switched off. Exit is just over there and there. No, I'm kidding. Please make sure they're on vibrate or mute, or just simply switch them off so that we don't disturb our very, very brave researchers. So now it's time for me, it's an absolute honor to give the floor to our first presenter today. It's Lieselot de Wachter, who is actually going to show and tell us what is it that she wants to do to break the wall of bacteria. Lieselot, you have the floor.
What is more dangerous, cancer or bacteria? This may seem like an obvious question, and for now you are right. Cancer is far more deadly than bacteria are. However, if we would come back here in 30 years, the answer will have changed. By 2050, more people will die of bacterial infections than of cancer. And this seems strange. After all, since the 1920s, we have developed many antibiotics that can easily kill bacteria. And these discoveries led to huge optimism. We were so sure that soon we would defeat all bacteria that we started paying less and less attention to them. As a result, the development of new antibiotics has been steadily declining since the 1980s. But we were overconfident. In reality, the fight was far from over. While we were looking the other way, bacteria started fighting back. They changed and evolved until they were no longer sensitive to the antibiotics we throw at them. These curves give you examples of specific bacterial species and how many of them have become resistant over time. Now, we just stood by and watched this happen. Today, the rise in antibiotic resistance, combined with the decrease in the development of new antibiotics, has brought us to the verge of a global health crisis. Soon, bacterial infections can no longer be treated with the antibiotics we have, and trivial illnesses will become deadly again. And this is why every day in the lab, I'm trying to find innovative and original ways to kill even the most resistant bacteria. Using exciting cutting-edge technologies, such as CRISPR genome editing and high-throughput single-cell microscopy, I'm trying to figure out how bacteria grow and which processes are important for them to survive. I want to identify these processes in the bacterial life cycle and find ways to block them. This way, I can stop bacteria from growing and make them die instead. And this is how I want to identify new antibiotic targets and tackle the looming antibiotic crisis. And I sincerely hope that that my work, will help to prevent bacteria from killing our future generations. Thank you.
Thank you very much, Lieselot, for actually breaking the ice. Are there any questions for Lieselot? No, in this case, thank you so much and congratulations for your research.
Thank you.
Next here with us is Bruno Rodinho Janeiro, who is going to talk to us about how he's actually breaking the walls around phage therapy. You have the floor.
Thank you very much. Do you know how many people will die because of the antibiotic resistance in 2050? It's estimated that 10 million. 10 million people will die annually and globally. And why? Because the antibiotic is the only weapon that we have, and the main weapon that we have to attack the infectious diseases, and they are stopped working. This is why we need to look for new tools, and one tool can be the viruses against the bacteria. These are called the phage, the bacteriophage, and these were used as therapy before. This is one example. This girl, this 15-year-old girl, has cystic fibrosis, and a chronic infection with Mycobacterium. The antibiotics didn't work, and the infection put her life at risk. But one research group isolated the bacteria, checked several phages against these bacteria, picked the best of them, and then prepare a cocktail. Treat these patients with this cocktail, and as you can see in the PET, the patient got cured with the treatment. But the problem is that the phage therapy, for the phage therapy, is that the phages are very specific. Can kill one bacteria, I cannot kill very closely related bacteria. This is a problem. If we can predict the effect of the phage before to use it, we can improve the phage therapy. And this is the objective of the project. But how we can do this? The phages and bacteria compete during millions of years. The phage try to kill the bacteria, and the bacteria try to survive this. The bacteria use the antiphage defense to do this. One way to try to systematically find this defense is to analyze the genome of the very closely related bacteria with different susceptibility to viruses, and then to identify the bacterial genes that produce the defense and the systems that the phage use to break this defense. This will be essential to do personalized and designed phage therapy and to improve the future of the phage therapy for the treating of infectious disease. Thank you very much.
Thank you very much, Bruno, and actually to filling the very, very difficult task of explaining a lot of complicated concepts in just 3 minutes. It's absolutely incredible. Are there any questions for Bruno? Yes, I see a question over there.
Yes, just a curiosity. Which phages are you working with?
In fact, we are looking for new phages, and this is another interesting point that we are looking in the hospital sewage to try to locate like to find new viruses against these clinical strains. In fact, we are focusing in very aggressive clinical strains and to try to isolate from 2 sources mainly, the hospital sewage and the stool from the patients.
Thank you very much, Bruno, and thank you for this question. Next on the stage, we have Tinta, who is going to tell us a little bit more about jellyfish degrading bacteria.
In recent decades, an increase in jellyfish blooms was recorded in several marine ecosystems around the globe. This can have serious ecological consequences, like affecting the global climate and healthy status of the ocean, but also socio-economical consequences. Because when jellyfish bloom, they can, for example, clog fishermen nets. When they bloom, they can block cooling intakes for coastal industries. And they can cause great damage to the coastal tourism. And I'm sure many of you were stung by a jellyfish. When jellyfish bloom, they can reach huge amount of biomass in a very short period of time. And when their population dies off, it does so en masse, meaning all together at the same time. This results in great amount of jellyfish carcasses and detritus in the ocean. Master recyclers of organic materials, such as carcasses of dead animals like jellyfish in the ocean, are in fact its tiniest inhabitants, bacteria. These are actually the most active, diverse, and numerous organisms in the ocean. And their major role is to decompose and degrade this complex organic material, like jellyfish carcasses are, back to the building blocks, making them available to other organisms in the ocean, like phytoplankton. In this way, bacteria in the ocean importantly affect global cycles of carbon and nitrogen, and thus the global climate, and are crucial for healthy functioning of marine ecosystems. But otherwise, in ocean, food supply is pretty scarce and very diluted. So we started to wonder what happens when all this huge amount of jellyfish detritus becomes available to bacteria. Because jellyfish might be 90% made out of water, but the rest is proteins. And this is superfood for some very specific bacteria, which might not be the same that we typically find in the ocean. So in our project, we investigate how, but also which bacteria can actually decompose this jellyfish detritus, and how is this affecting global cycles of carbon and nitrogen and healthy functioning of marine ecosystems. So by breaking the wall of jellyfish degrading bacteria, we hope to be able to better predict the implications of potentially increasing jellyfish blooms in the future for the health of the ocean and for the global climate change. Thank you.
Thank you very much for this incredible presentation. I see a question over there.
The increase of the populations of jellyfish can cause ocean ecosystem collapse. This is what I understood. Not collapse, damage. But the increase in the jellyfish population is already the result of ecosystem damage, isn't it?
It's true, but
I'm very sorry, but unfortunately we need to interrupt you. But
we can talk later. Thank you very much for your question.
Thank you. Thank you. I take the boo all to myself, not to anybody else. That's fine. That also shows us the importance of asking a question really, really straight to the point, but it also kind of puts a lot of momentum in between now and the coffee break when you can jump on all these researchers. I'd now also like to invite Francesco Forno on the stage so that he can tell us a little bit more about what is it that he does for breaking the wall of antipsychotics chain reaction. Francesca Forno, please.
So, as a researcher, sometimes we spend so much time, just put so much effort trying to find the treatment, the cure for a disease. And then at the end of the day, the side effects are just so devastating that people are just not using the drug. So let's talk about schizophrenia. Schizophrenia is a severe mental disorder which nowadays affecting 23 million people worldwide. And as I said, many efforts were done to find the treatment for schizophrenia. It's available, it's on the market. There are mainly two classes of antipsychotic drugs. One is the conventional class, and now we have the newest generation of antipsychotic drugs. They display greater effectiveness in the treatment of schizophrenia compared to the old-fashioned drugs. But unfortunately, patients undergoing the treatment are also found to develop side effect, metabolic side effect, in particular obesity and type 2 diabetes. So what happens? People are just leaving the treatment, worsening their psychiatric situation. That's not what we want. We want people to stay under treatment because we want to control their psychiatric situation, but we don't want them to gain weight and to feel uncomfortable with their life. We just want to provide them a normal life, even if they are schizophrenic patients. So what are we suggesting here? We want to find the cause of obesity and type 2 diabetes related to schizophrenic drugs. And how do we want to do it? We want to investigate the ER stress in the liver. ER stands for endoplasmic reticulum. Now, briefly, the endoplasmic reticulum is a small part of the cell which is involved in the folding of protein, and ER stress occurs when the load of protein entering the ER exceeds its folding capacity. Why did we decide to look at small ER stress in the liver? Because nowadays it's well known. Many studies have demonstrated that when ER stress occurs, especially in the liver, because of course the drug has a different mechanism of action in the brain and in the liver, it plays a key role in the development of chronic metabolic disease. And on the market there are relievers of ER stress. They have been approved for human use. So basically a combination between ER stress relievers and anti-schizophrenic drugs might be a possible treatment for schizophrenic patients. And I'm proud to say that actually with my project we found that one of the two main drugs I'm talking about, you can find it on the market under the name of Abilify, it's causing a huge ER stress, meaning that we are in the right direction, and with our research, we may be able to provide a better life to 23 million people all around the world. Thank you.
Thank you very much, Francesca, and congratulations for a fantastic presentation.
Thank you.
Now, just as a rule for us so that we can make sure that I do allow you a lot of time to actually get your questions answered, already when you see the question slides appearing, just already raise your hand. I'll see where you are, and then if there are any questions, then I'll just come here and interrupt our researcher so that we can hear your question. Thank you. Next on the stage, we have Fulvio Flamini, who is going to tell us what is it that he's doing with WALL-E.
Thank you. So this is a true story of courage and love. It is a story of two fields of research. The quest for artificial intelligence, or AI, with all its widespread and fascinating applications, and light-empowered technologies, such as lasers or fiber communication, which are fast and efficient. Yet, these two worlds are still far, and AI is still bounded to our electronics. So, what would happen if we brought these two fields together? What if we let AI liven up all our light-empowered equipment? The answer was given by the sci-fi film WALL-E by Pixar. WALL-E is a cute electronic robot on a future abandoned Earth who one day is visited by Eve, a female robot with a much more advanced and photonic technology. WALL-E falls in love, but eventually he risks to lose her due to low battery issues. The film criticizes consumerism, waste management, and human environmental impact. So we can actually learn from WALL-E where we are today and what more powerful and more energy-efficient solutions we need to get where we want to be. We just need an idea and the courage to pursue it. But as I said, this is a true story. Within my research, I describe a way to implement AI directly within photonic applications. And the intuition is actually really simple. Just like a message in a bottle follows the water flows, a photonic AI can learn and it can communicate by just looking at where particles of light propagate along waveguides. So the point is that now what's nice is that the required technology to achieve this task is already all available. And it can be integrated in a tiny piece of glass, which is as small as this one. So in the end, WALL-E reminds us that we should become smarter and smarter, and soon, to avoid a catastrophe. But until that point, with this technology, he and we will have new photonic processors that are really, really smart. Thank you.
Thank you very much, Fulvio, especially for showing that the photonic processors are the key to successful love relationships. Thank you very much for your presentation. The next presentation is going to be given by Ariadne Jebala Kukula, all for old-fashioned food testing. She's been driving me crazy. I've been making so many mistakes with her name. Ariadne, you have the floor.
Thank you very much. Have you ever wondered why are there so many food-related scandals? Why you might come across unwanted substances in your food? Substances like pesticide residues or allergens in a food labeled as allergen-free, or any other type of contaminant. Imagine the life of barley. It starts in the field where it grows, it harvests, and then it gets transferred into the industry where it gets transformed into a refreshing bottle of beer. During this time, a lot of tests are performed in order to reassure the quality of the final product and thus your safety. According to the EU legislation, those tests include screening using ligand-binding assays, and when the result of the screening is suspect, then confirmation is performed with analytical chemical tools involving mass spectrometry. But if so many tests are performed, the question remains, why are there still sometimes unwanted substances in the final product? The reason is because this whole procedure is time-consuming, and it's not very efficient, and it involves a lot of lab work. In our project, we tried to come up with a solution in order to add the efficiency that lacks from the current food testing strategy. And we're trying to combine the screening with the ligand binding assays and the confirmation in one line, and thus eliminating any extra steps of lab work. And in order to do so, we are using novel mass spectrometric techniques that can be used in ambient conditions. And in this way, we are emphasizing only in the advantages of both techniques while eliminating any drawback. However, this is something that has never been done before, and it's more easily said than done. And I'm trying to play the role of the mediator in order to bring you safer food. Thank you.
Thank you very much, Ariadne, for this presentation. Are there any questions? In this case, thank you and congratulations for your research. Next here with us, we have Harita Korlil. She's going to talk to us about chronic pain. Please, you have the floor.
Thank you. So I think all of us in this room can relate to this picture. It's a beautiful picture of a mom and daughter hugging. But you imagine if you have chronic pain, maybe neuropathic pain, this hug right there would be so painful that you wouldn't be able to enjoy it. 100 million Europeans suffer from chronic pain, different forms of it. And 100 million is a huge number. It's almost the entire population of Germany. And this actually has the chronic pain patient cannot do anything that a normal person does. Cannot sleep, cannot walk, cannot exercise, cannot do any household activities. And 38% of them say that they cannot manage their pain well. We do have existing treatments, but they still cannot do it. So what are we missing here? Why is it that you cannot manage the pain really well? If you look at the brain of a chronic pain patient, you can see that the brain rewires as the pain progresses. In simple words, whatever you're experiencing is constantly changing the structure and the function of your brain. This process is called brain plasticity or neuroplasticity. So to put this in perspective, I'm going to give you an example. So imagine the brain networks to the electrical wiring of your home. So now, if you're a healthy person, you want to turn the light on, and you put the switch on, and the light turns on. Now, for a chronic pain patient, the brain networks are messed up. So when you put the light on, the refrigerator is turned on, or the TV is turned on, or it's just mixed up. That's how complex it is in your brain. So we're trying to find out an answer to that question, and we already have a revolutionary technology called the functional ultrasound to see the brain in action. So what you see here is the entire vasculature of your brain. And using this technology, we can introduce the drug that we are currently using to treat the pain and see how it works in these networks. Can it really change the neuroplasticity? Can it really bring it back to normal? And to find out if the neuroplasticity from pain is reversible or not. You know, people, when they're in pain, they build these walls around themselves. It's a wall of helplessness. So today, let us pledge to break down that wall of suffocation and suffering and unhappiness and open doors to 1 million possibilities with effective pain management. Thank you.
Congratulations for your presentation, Harita, that can have a huge impact on a lot of Europeans. Are there any questions? We have 10 more seconds. 9, 8. Thank you so much.
Thank you so much.
Next here with us on the stage, we will have Benjamin Raymond, who will actually show us how is it that he's breaking the walls of tuberculosis.
Thank you. Now, not many people here today might realize this, but evidence from prehistoric humans have shown evidence of infection with tuberculosis, as have mummies from ancient Egypt. This long history of tuberculosis leads us to today, where it still persists as a globally devastating disease. 2 billion people are currently infected with tuberculosis. That's almost 1/3 of the world's population. Just take a second to think about that. Sadly, 1.8 million people die from tuberculosis every year. Now, tuberculosis is caused by the bacteria, Mycobacterium tuberculosis, that colonizes our lung, where it is very adept at evading our immune system and causing very significant inflammation. Now, one of the major problems is that the treatment for tuberculosis requires an intense and prolonged course of antibiotics that does not even guarantee that the patient will be cured. Additionally, the most widely used vaccines today only offer limited protection from the disease. Therefore, at my institute, we are working towards better therapies to fight against tuberculosis, specifically via identifying novel therapeutic targets. And in order to do that, we need to better understand the interactions between the bacteria and us. Now, upon contact with the lung, tuberculosis induces a myriad of different immune responses that are mediated by immune cells. And one such response that I'd like you to pay particular attention to is called the interferon response. And to put this simply, this interferon response signals to the cells of your body to tell them to start fighting infection. And thus, this plays a very crucial role in the outcome of tuberculosis. However, as with most things, this isn't so simple, mainly because too much of this interferon response can actually damage the cells of our body and actually aid the progression of tuberculosis. So, we need ways in which we can modulate this response and to better understand the mechanisms involved in maintaining this fine-tuned response. And recently, we've identified one such factor, a sensor expressed in numerous cells of the immune system that we refer to as DCIR. What we've recently shown is that DCIR plays a crucial role in regulating our immune response to fight tuberculosis via this interferon pathway. And so what I've done recently is that I've identified the specific molecule that DCIR interacts with, and I want to expand upon this discovery by looking at how this interaction plays into the interferon response. I want to further this and to develop molecular tools that can be used to either stimulate or inhibit DCIR in the hopes of regulating our immune response so that we can finally work towards breaking down the wall of this ancient disease that has plagued humanity for millennia. Thank you.
Thank you, Benjamin. It's absolutely incredible what you're doing. Are there any questions for Benjamin and his research? If not, thank you and congratulations.
Sure.
Next, and already mic'd up and prepared, we have Thomas Winkler, who is going to tell us about how he's breaking the walls of brain's blood vessels, literally. Thomas, you have the floor.
Every second person in the EU is living with a brain-related disorder, like Parkinson's, bipolar, or brain cancer. That's a higher percentage than for any other organ. So why is drug development in this realm so slow and actually slowing down? Well, it's not enough for a successful drug to show therapeutic effects on the neural cells in the brain. The hundreds of kilometers of blood vessels in the brain are lined with cells that form a literal wall. This wall, that we call the blood-brain barrier, protects the brain from disease. But it also means that drugs can have a hard time breaking through. Now, we can't really study this complex interplay between neural cells and brain blood vessels in live humans. We generally do not like our brains sliced into. So instead, both academic research and drug development have to rely on stand-in models. Typical cell culture models typically look at one or two cell types in a plastic container. But our brain is much more complex, of course. Lab rat or mouse models certainly have a lot of complex biological interactions, but they're not necessarily the same ones as for us humans. This is where my project comes in, with a new generation model that I termed the NeuroView. It combines for the first time 3 advantages to give us a better stand-in for the human blood-brain barrier. First, we rely on stem cells that we guide to form blood-brain barrier cells to get us very human-like biological function. Second, the plastic microfluidics allow us to provide each of these cells their preferred environment, much more brain-like than a typical plastic container. Last but not least, we can continuously monitor what's going on inside the NeuroView. With these key advantages, we provide a next-generation model system that can allow us to reduce animal testing, so our favorite lab rat can get back to world domination. And So drug development can accelerate again in this realm. This is the first step toward new cures and treatments for those suffering with the likes of Alzheimer's or schizophrenia. Thank you.
Thank you very much, Thomas. Are there any questions about his project, New Review, or a new world domination? No? Thank you very much, and congratulations for all your research and your presentation. After Thomas, we have Rita Branco. Rita is going to tell us about how she's breaking the wall of groundwater pollution.
Thank you. Okay. So, good afternoon. By show of hands, who in this room drinks water? A lot of people. We are 7.7 billion people in this planet, and we all have to drink water. And now, who knows where drinking water comes from? Well, 50% of it comes from one single source, groundwater. But there is a problem with groundwater, and it's similar to the problem of the water inside this bottle. It looks clean, you'll probably even drink it, but it's actually polluted. Just like this water, groundwater around the world contains micropollutants, like pesticides and pharmaceuticals, present at very low concentrations. For example, a study that analyzed 160 European groundwater samples detected pesticides above the limits set by the European Union in 29% of the cases. And this is a big problem for drinking water companies that need to remove micropollutants in their drinking water treatment plants. And the technologies used also have problems. They are costly and can generate toxic byproducts. So we need to find a sustainable and cost-effective solution to ensure that our water is clean and safe. And this solution can come from nature. There are microorganisms, like bacteria, naturally present in groundwater that can degrade micropollutants. Unfortunately, this process is very, very slow. But maybe there is something we can do to boost it. Laboratory studies have shown that the addition of dissolved organic matter can increase the rate of micropollutant biodegradation by microorganisms. So, in this bottle, I have dissolved organic matter, and this can easily be obtained from compost, something natural that we can even produce at our homes. So, we can inject this into groundwater using existing wells, and then groundwater flow would do the job of mixing dissolved organic matter, micropollutants, and microorganisms all together, creating this reactive zone where the degradation of micropollutants occurs faster, so that in the end, when groundwater reaches the extraction wells, all the micropollutants have been degraded. And this is how, without the need of new infrastructures and using only natural resources, we plan to break the wall of groundwater pollution, so that in the future, we can all relax and drink groundwater freely, knowing that nature has our back. Thank you.
Congratulations, Rita. I hope that the organizers of this event have passed by your place so that you can check the water that is being served. If not, they should definitely do it now. Are there any questions for Rita? Yes, please.
Micropollutants problem is only for groundwater or generally for drinking water?
General for drinking water, yeah. I'm focused more on groundwater because I'm doing my research in the Netherlands, and there 65% of the drinking water comes from groundwater. And actually, in the European Union, the number is higher, is 75%. So this is the future, we think, and that's why we are focusing on it.
Thank you very much, both for the question.
Thank you for the question, sorry.
And thank you, Rita.
Thank you.
Congratulations.
Thanks.
Next and all prepared is Benjamin Droguet, who is going to tell us a little bit more about polluting colorants and microplastics.
Is it really what surfing is going to be like in a few decades? That's really sad, right? And this is only what you can see with your eyes. According to the UN, there are more than 51 trillion pieces of microplastic particle in the seas, which is 500 times more than stars in our galaxy. And these microplastic particles can be quite small, and therefore, until living organisms eventually accumulate and harm them and us. Among these, more than 200 billion pieces come from the use of personal care products with intentionally added microbits. Several EU countries have recently banned the use of this product with exfoliating beads, for instance, shampoo, shower gel, and toothpaste with such beads. But these current laws ignore all the other use and type of microplastic, including a major one, plastic glitter. Hence, does it mean that the party will soon be over when it comes to glitter? Well, nature gives us the solution. This fruit has one of the brightest colors of all biological material on Earth. And surprisingly, the color that we see doesn't come from any pigment nor dye, but instead from a nanostructure of cellulose fibers that are able to interact with light in order to produce this brilliant blue color. In our lab, we have replicated such structure in astonishing color using cellulose extracted from wood to produce 100% biodegradable glitter and pigment. And by simply engineering the nanostructures, we are able to produce all the color of the rainbow, even the most sought-after one, like gold. And the color can remain vivid for as long as the structure is preserved, which can be weeks to centuries. And yet, it is fully biodegradable. So nature knows how to produce striking visual appearance without generating waste. So instead of banning glitter due to the harmful impact it has on the environment, let's allow the next generation to join the party and glow responsibly for the many festive years to come with structurally colored pigment and glitter made from cellulose. Thank you.
Merci, Benjamin, and I also hope that the closing event will also be checked by you to make sure everything is sustainable and fun. Are there any questions for Benjamin? If not, thank you so much for your presentation and for your work. Next is Chandril's turn. Chandril Gandan Ghosh is going to talk to us about how he's breaking the walls of mental ill health.
Hi. 3 years back, when I lost my best friend to suicide, it changed my life forever. She was diagnosed with major depressive disorder, and she was on antidepressant pills. I still remember she used to tell me that none of these pills are helping her. And as a clinician, I have seen a lot of patients do not respond well to conventional treatment plans. So what is going wrong? Is it the pharmaceutical industry engaging in unfair trade practices? Or is it the doctors who are making the mistake in prescribing the wrong medication? Well, it can be both. But not all doctors make mistakes all the time. Neither all the pharmaceutical companies engage in unethical practices either. So is there something we are missing out? We have all heard terms like bipolar disorder, major depressive disorder, schizophrenia, and so on and so forth, and drugs being developed on this. But what if, like the research studies have repeatedly demonstrated, these disorders might not even exist? What if the way we conceptualize mental illness was wrong? Well, manuals which tried to conceptualize mental illness were created primarily based on the clinician's personal experiences and their observations of the patients, and not the research data as such. The initial ones and the later ones which got developed were based on primarily on this, which ultimately led to the diagnostic manuals not just unscientific, but it also misled the pharmaceutical industry into developing medications for disorders which does not even exist. So, instead of focusing on or taking the clinician's observations of the patient's experience, why not build a manual directly based on patients' lived experiences? And that's exactly what I'm working on. So I'm working on a cloud-based manual that will hear the patients, their experiences, and with the artificial intelligence it is equipped with, it will figure out the kinds of patterns within it.
Yeah.
take a question just from there.
All
please.
right.
Thank you very much. I
Thank
have
you.
a question.
Sure.
What is for you the difference between a mental disease and a brain disease?
All right. Thank you so much for the question. Can we catch, because the answer will take long. Okay, can we meet at the coffee break? Thank you. Thank you so much.
I would call this the nicest way of getting another possible funder for your project. Please come back and tell us how is it
Sure.
this work. Congratulations for
Thank
your
you so
work
much.
and for showing us how else is it that we can look and tackle mental illness.
Thank you so much. Thank you.
Athanasios Rogdaikis is going to tell us how he's tackling the wall of dementia. Athanasios, take it from here.
Imagine you wake up in a room and everything around you feels unfamiliar. Strangers act like they know you and love you, but you have no idea who they are. You feel scared, confused, alone. This is how a typical morning feels for a person suffering from dementia, and it could happen to any of us. At the moment, there are 50 million people suffering from the disease around the globe. This number will raise to 150 million people by 2050. The most common form of dementia is Alzheimer's disease, and accounts for almost 70% of the cases. Its pathological features are attributed to two proteins which behave abnormally, and they form aggregates inside and outside the cells. These aggregates then cause neuronal cells to malfunction or even die. Neurotrophins, on the other hand, are a family of proteins that are physiologically produced in the brain. Their name comes from the Greek words for neuron and food. Essentially, they are proteins that feed the neurons and have neuroprotective capacities. In addition to that, it has been shown that in Alzheimer's disease, neurotrophins production and action is reduced. However, there are limitations in their use as drugs that are mainly attributed to their size. Being large proteins makes it difficult to reach and navigate the brain. Our solution to this problem is called uroneurotrophin. We are a team of researchers, and our goal is to develop small molecules which can mimic neurotrophin action and can be used as drugs to tackle diseases like Alzheimer's. As a model molecule, we use a steroid hormone that is abundant in the brain, and it's called DHEA. The cool thing about DHEA is that it mimics neurotrophins, it is a small molecule, and can protect neurons from dying. The not so cool thing about it is that it has various side effects, including cancers. Nevertheless, by slightly tweaking its structure, we can improve the neuroprotective capacity while at the same time limit its side effects. Therefore, the workflow of our project looks a bit like this. We model the action of new hypothesized compounds. We then synthesize these compounds and assess their biological activities. Depending on the result, we come up with improved models that will lead to the synthesis of better compounds. Our ultimate goal is to develop the one drug that will break down the wall of dementia. Thank you.
Ευχαριστώ, Athanasios. Is there anybody that would like to take the challenge of asking a question? Yes, please. Quickly over there.
What is the best candidate you have simulating neurotrophins?
We have developed a small molecule. We term it BNN27. This is already published, and we're trying to work on even better compounds that will fight Alzheimer's disease. Thank you very much.
This is a new strategy. Well done, Athanasios. Next here with us on the stage, we have Nicolás Robinson Garcia, who is going to break the wall of science policy.
Thank you. Yeah. A shape with 5 legs is normally what we're looking for often in science. Talented individuals who excel in all the different tasks that they are supposed to do. And we assess them in an isolated way, ignoring that part of their achievements are actually thanks to supporting authors and scientists who are working with them and collaborating with them. These types of researchers can be technicians in labs, statisticians, computer scientists working in biomedical teams also, who have not this leading position in their role. The objective of my project is to identify this diversity of profiles in research to be able to ensure and inform policymakers on better ways to design career choices for scientists, and not only have this homogeneous way of assessing their outputs. For this, we're working with a set of publications in which we have information on the type of contributions that researchers are doing in each of the papers, and combining this information with bibliographic variables, and using this data set as a way of training, using a machine learning technique, to be able to infer the contributions of these researchers in each and every one of the publications that they've had during their whole career history. The goal is to identify how at different stages in their careers they have different profiles, and also within each of the stage there are different types of researchers doing different things. So for instance, we do find highly productive researchers with high impactful publications which mainly perform analysis but do not conceive and design the studies that they are performing, or other individuals who are actually contributing with tools but not doing the rest of the tasks, or just conceiving and designing and managing researchers but not actually performing the experiments. We hope that the results of this study will show that actually a ship with 5 legs is a dysfunctional ship, and that we need this type of teams actually to make science more efficient. In this room, we have a group of very talented researchers who want to lead their own projects. And in order for them to achieve it, we also need to give the tools for their supporting authors and scientists who are going to help them achieve this, to have their own career choices and be able to ensure that this talent is not lost from academia. Thank you very much.
Thank you. Are there any questions for Nicolas? If not, thank you very much for busting the myth around science policy.
Thank you.
And last person in this first batch of courageous change agents is Mariana. Mariana Perez-Ibarech is going to talk to us about resistant microorganisms. Please, Mariana, you have the floor.
Thank you. Can you believe that a tiny and invisible microorganism can kill all the people in the world? Let me show you the 10 years challenge of the bacteria. The white circles are the antibiotics. The cloudy parts are bacteria. The clear zone around the antibiotics is the zone where the bacteria can't grow for the presence of the antibiotics. 10 years later, the same antibiotics, the same bacteria, less side of the clear zone, where now the bacteria can grow, even though the presence of the antibiotics. Now, imagining this same bacterium, but infecting our body. Probably, with these things, we will die. But do you know how it's born? How many times did you not finish an antibiotic treatment? How many times did you self-medicate? When that happens, the microorganism can generate mutation in its DNA, becoming in a super bug. Then, this DNA can be shared with another population, increasing more and more the number of microorganisms, super microorganisms around the world. The problem is so serious that if we don't work quickly, we are near to enter in the post-antibiotic era, where just a simple cut in your fingers and regular practice could be potentially deadly. Our proposed is the use of antimicrobial peptides, especially the use of the niacin, which is a little protein produced by a good bacteria and has the ability to kill or inhibit another bacteria. However, some of these bad bacteria that we want to kill have resistance against this niacin and prevent its action, cutting it in two or just expelling it out the cells. Our proposed is the use of genetics engineering to create camouflaged niacin, which cannot be recognized for the microorganisms, and in this way, it's able to kill it. Nowadays, we already obtain neoniacin with an enhancing inhibition activity up to 20 times compared with the use of regular niacin. This project is still under development and not ready to be used in humans, but it's offers hope to win the war against resistant microorganisms. Thank you very much.
Thank you, Mariana. Are there any questions for Mariana? Yes, please. I see first question over there.
Do you consider this war against bacteria winnable? Because based on current evidence until now, it seems we're in the direction of losing it, and we still keep technology for fighting it.
If we are able to win the war, you say. Yes, I think we can. There are a lot of people around the world looking for different kind of solution against the resistant microorganisms. So I think we'll be able to win the war.
Yes, Mariana, thank you so much, and thank you for scaring all
you.
of us for doing self-doctoring online. Thank you. You have now seen the 15 first researchers, and I would kindly ask you to give them a super, super warm round of applause. As our best friend over here is telling us, we have 30 minutes of a coffee break, so see you back here at 3 p.m. Thank you. Ladies and gentlemen, welcome back from this very interesting break where I hope a lot of connections have already been established. There were a lot of questions for our researchers, therefore a lot of interest in their scientific projects. Before we go in the second batch of presentations, I would like to remind you of the fact that you do have a very, very important responsibility, and that is to also vote for your favorite researcher. So what I have here is the voting ballot that I'm hoping everybody has received as you entered. If not, I have colleagues just there at the back that can hand them out for you. Here on the back, please write down the name of the researcher that you would like to vote for, and at the end, please give them to the members of staff just at the back. As a quick reminder, after the second batch of presentations, do not leave the room because we're going to discuss a little bit more about Maria Skłodowska Curie actions, and also about the Falling Walls lab, and we're also going to hand out several certificates to all the participants that we have today. So just give these ballots at the end, and please stay in the room with us for about half an hour of discussions. I was thinking, I was quite inspired by Fulvio's presentation, and I actually realized I do have a joke about a photon. So a photon goes into a hotel and is being immediately asked, "Gentlemen, would you need any help with your luggage?" And the photon answers, "No, I'm traveling light." So with this in mind, oh, thank you, thank you. I've worked a lot for this, I've worked a lot. Because you haven't come here to actually listen to me, but to listen to absolutely very, very important and the change agents for tomorrow. I'd like to invite here on stage Cristina Makundu, who is going to talk to us about how she's breaking the walls on vulnerable users' injuries. Cristina,
Thank you.
have the floor.
By show of hands, who of you crossed the street today walking? And who used the bike to get from a point A to B? Yes, those actions are natural. In fact, with the increase of the population of all journeys, up to 40% are traveled by cycle or on foot. But those users are vulnerable, more subject to accidents, to fall on road. I'm sure that you can visualize this collision accident between a car and a cyclist or a pedestrian. From all the injuries caused by the accident, 30% are caused by the fall on the road. We call that surface impact. And surface impact is important. And that can mean that you will be disabled and not be able to walk anymore or to play with your children. How do you think we can inverse this tendency? Here, my project comes with the objective to create an innovative pavement, soft, but able to support the daily traffic, and at the same time reduce injuries by absorbing this surface impact. But how? Using rubber. Thanks to its property, elastic property, we can use rubber in order to make the pavement softer. The plus? I do not use technical rubber produced just for the research. I use recycled rubber coming from end-of-life tires at high percentage inside of my mixtures. This improves the life cycle of the tires and reduces waste. As a chemist, my challenge is to deal with the porousness of the rubber mostly, and to make the formulation works, and at the same time durable and costless. I bring some samples. This is the pavement you can find in the streets now, and this is one of my samples. You can see they are similar. This is encouraging, but the success will be the paving of this new material and the collection of number of data about the number of lives saved. Instead of paving roads that are perfect for all the users, just trucks or cars, let's make roads that are perfect for all the users, including vulnerable users. Prevent unnecessary pain and tears. It's a new horizon, but a safer one. Thank you.
Thank you. Thank you very much, Christina, for challenging road infrastructure.
Thank you.
If there are no questions for Christina, thank you.
Thank you.
I'd like to invite here to join us Ivana Cavaliere. She's going to talk about breaking walls for Parkinson's disease.
Good afternoon to everyone. Nowadays, more than 10 million people worldwide are living with Parkinson's, and their quality of life is deeply compromised by several motor dysfunctions. One of the main characteristics of Parkinson's is the progressive decrease in the level of dopamine, a neurotransmitter that plays a crucial role in the motor control. Obviously, the best treatment will be the replacement of the lost dopamine. However, it is not able to reach the brain because of the presence of the so-called blood-brain barrier, a wall made by different kinds of cells that isolate the brain. For this reason, current therapies are based on the use of levodopa, a precursor of dopamine, and nanoparticles that can be loaded with dopamine, and they both can cross the blood-brain barrier. However, these solutions are not optimal, presenting many disadvantages and producing side effects. One encouraging approach is not to cross the blood-brain barrier, but to overcome it by administering drugs through the nose. A nasal spray. I don't have it now, but you can simply imagine it, like one that you buy in pharmacy for a simple cold, but for Parkinson's disease. However, it still doesn't exist for dopamine. So, our idea is to combine the advantages of nanoparticles with the intranasal administration, the nasal spray use, in order to obtain a new drug and treatment that will increase the therapy efficiency and break the wall of dopamine administration. Our promising nanoparticles are mainly based by dopamine and iron and obtained by a very simple reaction. These nanoparticles possess a very high amount of dopamine, which is translated in a reduction of the daily dose. This system is made by potentially not toxic molecules, avoiding other damages to the organism. The small size is a crucial parameter to reach the brain and for the comfort of the patient during the administration. Iron can act as a contrast agent in MRI, so this system will offer even diagnostic capabilities, helping to monitor the disease. Most importantly, these nanoparticles have shown to be able to ameliorate symptoms of Parkinson's during intranasal administration in vivo. Clearly, the next step will be to test them in humans, but we strongly believe that this drug and treatment would really and concretely help patients affected by Parkinson's disease. Thank you.
Are there any questions for Ivana and her breakthrough research? If not, thank you very much, Ivana, for your presentation. Next with us is José Esteban Rodriguez Condia, who is going to talk to us about breaking the walls on devices reliability in autonomous cars.
Thank you.
To the future.
Well, let's imagine that you live in a world where the cities and roads are crowded with self-driving cars. And now let's imagine that every morning you wake up, you enter a self-driving car, you maybe drink your coffee, you read the newspapers, and you go directly to your work. Even if you have enough time, you can even work a bit. Now, this could be your future in 20 years from now. However, I have some bad news for you. Still, the chips, in this moment, are not able to provide enough mechanism in order to be able to work for long-time executions. Chips like this will be failing after long periods of execution, and these chips will be included in a self-driving car. What they do? These particular elements are much more smarter and much more faster than ever before. However, now they are much more prone to faults. And this sensitivity to faults is make them fail after long periods of execution by aging effects or by interactions with the environment. Problems related with change in temperature, for instance, or maybe the radiation exposure could cause of them to cause this kind of misbehaviors. And one particular misbehavior, for instance, in detection, could generate a really critical consequence. For instance, a death or an injury of a passenger or maybe a pedestrian. We don't want that. If you will be using or you will buy a self-driving car, we want you to be safe than ever before. My solution is to provide in-field techniques that should be providing a particular shield for this kind of chips. So basically, I have to re-engineering at multiple levels, hardware and software, who can I plan to develop the particular chip and how can I protect chips before to detect. Second, to mitigate, and third, to be sure that even if you are able to detect and mitigate the faults, the final functionality of the chip will be present and will be continuing working. At the end, we plan that, the main idea is that you, after 10 years, 20 years from now, you will be able to drink your coffee and be sure that the best technology ever and the best reliability techniques were used to make your chips and to make your self-driving car working better. And thank you very much.
Thank you for your very timely presentation and a lot of organizations that would definitely like your feedback. Are there any questions? If not, thank you so much.
Thank you.
Next here with us is Ivan Palaya. He's going to break the wall of cement.
Imagine a truck that every year for your birthday delivers to you half a ton of cement powder. It's exactly that, 500 kilograms, the quantity of cement produced every year for every one of us. With it, we build our houses, our towns, this building, the canal. Imagine the impact of that on the environment. Well, cement production is responsible for up to 10% of our CO2 emissions in the atmosphere, way more than planes. Cement is the most produced substance in the world. And yet, nobody really knows how it works, why this building does not collapse. My ambition is to answer that question. And to do so, I need to look at what happens in cement at a nanoscopic scale. I'm a theorist. I don't even know how to use a microscope, but I built my own little cube of concrete on my computer. I spend hours and hours analyzing it, and then I translate what I learn into equations that scientists around the world will be able to use. Our common aim is to build a stronger and greener cement. Let me tell you what I learned. To do so, I need to tell you a love story. A wonderful romance taking place in a cement mixer. It's the impossible story of two identical cement particles, the blue stuff, both negatively charged. As you know, in nature, two negative objects repel each other. Well, not in cement. We discovered that these two negative particles actually stick together thanks to the presence of the small positive ions walking around in our cement mixer. These ions place themselves between our two cement particles and, we must say it, cement their relationship. They act like glue, like good mutual friends between two partners, or, to keep our feet on the ground, like a 20-year mortgage to share with your loved one, an unbreakable bond. We have to thank this unbreakable bond if cement is the strongest glue we know, if this building is still standing since I've started talking. One day, we will develop a new substance that works in the same way as traditional cement, but much less polluting to produce. We will then have the green cement with which we will build our houses, our towns, the canals of our future. Thank you.
Thank you for making us feel much more comfortable both in this building as well as in our very well cemented relationships. Are there any questions for Ivan? If not, thank you for a very strong presentation.
Thank you.
The stage we have Mafalda Ferreira, who is going to talk to us about how she's breaking the wall of lung cancer with liquid biopsies. Please, Mafalda.
Thank you. One of the main problems in cancer is being detected in a late stage, when it's already spread through the body. This means in a lung cancer patient, this patient has 6% chances to live 5 years. When we can detect this cancer earlier, this 6% goes up to 60%. This is a huge improvement in the survival of patients. When a tumor is detected, a biopsy is performed. This biopsy, removing a piece of the tumor, gives precious information to the medical doctors to select the right treatment for this patient. But this technique is invasive, uncomfortable, and cannot be repeated during treatment. What if we could do this in a non-invasive way by collecting a tube of blood. This is what we call liquid biopsies. The tumors release to the body, to the blood vessel signals, and we can get these signals in the blood. I'm part of the European Liquid Biopsies Academy, ELBA, and we are trying to detect these signals and decode this just like a barcode. In Amsterdam, in my team, we are testing the blood platelets for the liquid biopsies. We know that in cancer patients, this barcode is different than in healthy patients and in healthy donors. Screening these barcodes, we can find a way to detect cancer earlier, save more lives, and finally break the wall of cancer. Thank you.
Thank you, Mafalda, for your amazing work in such an important field. I see a question over there.
Hi.
Hi. So is this method applicable during treatment?
Yes, because we can take blood from the patient several times, so we can follow the treatment and collect the blood and see the signals in the blood, and following the treatment and adjust the treatment.
So it's also a localized signal.
Yes, because the tumors release signals to the blood all the time. Even before they metastasize, they go through other bodies, to other organs. Thank you.
Thank you as well. Are there any other questions? If not, thank you very much and congratulations
Thank you.
for your work. Thanks, Nahal. Next person here on the stage is Gillian Bratt. She is going to talk to us about her work in breaking the wall of blood vessel for cancer detection.
Thank you. Today, I would like to share with you a story, and this is the story of Diane. Diane is 35 years old, and she lives in Norway together with her husband Lars and their 5-year-old daughter Naomi. They've lived a very happy and active life until 4 years ago. Diane got diagnosed with an advanced stage lung cancer. This is currently still a death sentence. There's no cure for advanced stage lung cancer. I want you to realize that this could have been any one of us. Cancer is not a fair disease. It doesn't only pick people who are smoking or who are more elderly. Diane's challenge is to survive for as long as possible, to see her daughter grow up, to be with her husband. This number here represents our challenge. Every year, 1.8 million people die from lung cancer. Every year, 1.8 million people that have received that same diagnosis, that same death sentence. So why is lung cancer so deadly? There's two main problems. One of them is that we find the tumors too late, when there is no cure anymore. The second problem is we need something to decide which treatment to give to which patient. As Mafalda previously explained to you, it's very complicated to get tissue from the lungs, if not impossible. Even if we can do that, you can imagine that a tumor is very different on this side than on this side. So how do you get the full picture? This is what we're trying to do currently. We're breaking the walls of blood vessels. Tumors are like any one of us. We all need oxygen, we all need food, and we take that from the blood. While we do that, the tumors also release parts of themselves into that same blood. And we can use that. And we can use it for two things. We can use it to screen healthy people, like you and me. But also we can use it for the patients that are currently on treatment, to select the right treatment for each patient. I'm part of a multidisciplinary cohort, including medical doctors, scientists, and bioinformatics. And together we formed the European Liquid Biopsies Academy, or ELBA in short. This, what you can see here ongoing, is our goal. We want to use liquid biopsies. And we want to bring that number down, that 1.8 million, to prevent you and me and all the people that we love from getting that same death sentence that Diane got 4 years ago. If there's no questions, I would like to continue to thank Diane and Lars for letting me share their very personal story here with you today. And also my bosses, I'm a scientist, but they are medical doctors, and they've shown me a different side of looking at doing research in the lab. Instead of a bunch of cells growing, I see that they came from a patient at some point. Thank you. Thank you.
Thank you very much, Gillian, for this presentation. Next with us is Mercedes Regadio. She's going to talk to us about how she's breaking the walls on pollution and residues. Mercedes, please.
Did you know that we produce more than 2 billion tons of waste per year, and this is expected to increase by 70%? Where will all this waste go to? Landfills. 70% of the waste is disposed of in some kind of landfills. The problem of landfills is that it generates a polluted liquid, the leachate. So, to protect human health and the environment, barriers should be installed. Compacted clay liners are normally used because of their low cost and their high attenuation capacity. But they can swell and shrink, resulting in cracks through which pollutants leak. We can also use synthetic plastic liners, but they are expensive and they can fail in less than 10 years. My research is looking for a solution in 2 ways. First, can we add other materials to improve the performance of clay liners? For that, I'm using residues produced as a result of industrial activities, because they are free of cost and available in large quantities. Second, can we prove the performance of these new liners? At the University of Sheffield, I'm working towards proving the long-term performance over 100 years of the world's first recycled liner. Let me explain you how. We have the real scenario on the left, where landfills are operating for 25 years with a landfill liquid of 1 meter head and with a barrier of 2 meters thickness. We need to decrease the time from years to days and the size from meters to centimeters to be able to work in the lab. This can be done using increasing the gravity, which is represented here by the red arrow. And we are doing this with a geotechnical centrifuge, which is nothing else than a large spinning device. By rotating at high speed, we are increasing the gravity. This will decrease the experimental time and reduces the size of the experiment. Eventually, we hope to decrease the pollution risk of the waste, the disposal of residues by recycling them, decrease the cost of landfilling, and save a natural source like clays. Thank you.
Thank you for a very timely presentation as well as a very timely research on this matter. Are there any questions for Mercedes? If not, thank you so much, and good luck for the rest of your research on this. Next, I would like to invite Emmanuel Salifou, who is going to tell us about how he's breaking the walls of unsustainable soil laws.
In the next 5 seconds, the equivalent of a soccer field of soil would have eroded globally. And guess what? It takes up to 1,000 years to create just 3 centimeters of topsoil. The importance of soil becomes even more relevant when we realize that soil erosion is directly linked to hunger and starvation, to land degradation, landslides, floods, and water pollution. And none of us here is spared any of these consequences. And it gets worse with climate change. Indeed, we're carelessly losing an invaluable resource from under our feet and under our watch. The wall that I have come to break cuts deep into the ground. It's down to earth. But I propose to shatter it using mushrooms. Yes, mushrooms. See, soil erosion occurs when the impacts of water and wind causes the detachment, the transport, the deposition, and translocation of soil particles. So the solution is to improve the soil properties to be able to resist these erosive forces. So engineers typically use cement, concrete, and other chemicals which are strong and reliable, but whose production and installation processes are costly. They damage the ground, But more importantly, they contribute to carbon emissions, which causes global warming and climate change. And so we need better alternatives, like natural mushrooms. Do you know that 3/4 of the total microbial biomass in the soil is fungi? And that the single species of fungus is the world's largest and oldest organism, occupying an area that is 20 times as large as the Vatican City. Mushroom is fungus. And the roots of mushroom, called mycelium, can form vast 3-dimensional networks in soils and release natural chemicals that are significant for soil engineering. At the laboratory scale, we have developed methods and techniques to use these mycelium networks of oyster mushroom in sands to use them to improve the hydraulic and mechanical properties of the soil. This has been tested for soil erosion by comparing the volume of soils that are eroded when we have mycelium and when we have no mycelium. Up to 80% reduction in the soil loss has been achieved. If a single species of fungus can achieve this, imagine what thousands of other species could do. To overcome the devastating consequences of soil erosion, we must cooperate with the ancient fungal networks under our feet in order to preserve these invaluable soil resources that are under our watch. We owe this to ourselves and to the future generations. I thank you.
I see a question raised over there.
Hello. Yeah, deforestation is also a big factor in soil erosion, I think.
Yeah.
Do your fungi not interfere with the growth of new trees?
New trees? No. Well, ecologically speaking, fungi is at the beginning of life. So typically where you have no life at all, it's the growth of fungus that brings other organisms to even start growing at all. So it's not going to affect the growth of trees. We can talk about it more later.
Thank you.
Thanks.
Thank you for a great question, and I'm sorry I need to be the time responsible and cut the discussion, but I hope you can ask him this question also after the event. Next with us is Wojciech Rzadkowski, who is going to tell us a little bit more about how is it that he's breaking the walls of complexity. Please.
We all in this room believe that we are just humans. That is, of course, true, but each of us is also something else. You are a quantum many-body system, you are a quantum many-body system, and I'm also a quantum many-body system. In fact, everything in this universe which is larger than an elementary particle is a quantum many-body system. But let's take a very specific example. A quantum computer. We put great hopes in quantum computers, and we think that they will help us solve problems such as drug discovery or such as designing self-driving cars that would not collide. However, before we build a quantum computer, we need to model it, we need to do research on that, and for that we need numerical modeling. And as every quantum computer consists of bits, and we need to model these systems in terms of numbers. And then it starts gently. For 1 bit, you need just 2 parameters, 2 numbers to model it. But then for 10, you already need 1,000. And guess, for 50, you need 1 quadrillion of numbers. This is absolutely impossible to model at such scale. We need something much more efficient. And this is where artificial intelligence and recent developments in computer science comes in. So imagine that we can use neural networks, the same techniques that are used, for example, to tell apart cats and dogs by Facebook, can help us actually solve the problems of modeling of quantum computers. We can model quantum systems as neural networks. And then, again, we start very gently with just 2 numbers for 1 bit of quantum computer. Then it goes even higher than it went before, but let's see what happens at 10. At 10, it's already just 200 parameters. And then, for 50 quantum bits, we need just 5,000 parameters. Do you still remember? We needed 1 quadrillion at this stage previously without this artificial intelligence. Now it's a complete game changer. Moreover, we can use the same techniques that computer scientists use to optimize their networks to find the parameters that correspond to quantum states of interest. And this is how we tackle the cures of quantum complexity. Thank you very much.
Thank you. Are there any questions? If not, I would like to personally thank you for allowing me to come up with a very nice line whenever I need to explain that something that somebody said is complicated. I'll say, you're a quantum antibody system. So thank you for that. And congratulations and well done on your presentation and your research. I would now like to invite on stage Julia. Julia Zubadek is going to talk to us about inflammation makers monitoring and how she's breaking those walls. Please, Julia.
Thank you. So inflammation is actually part of the reaction of our body to a harmful stimuli, actually signalize that something is going wrong. However, when we are dealing with a silent, low-grade chronic inflammation, then this makes our immune system gets weaker and weaker, and that leads to diseases, illnesses, rapid aging, and serious diseases like, for example, cancer. That makes actually the inflammation a secret killer and also a serial killer. What we need to do is to be able to detect, reveal, and monitor the inflammation, hidden also inflammation at very early stages. So something can be done and the treatment can be applicable. What I'm suggesting is, and I'm also developing, is a wearable, miniaturized, ultra-sensitive platform, sensing platform that will offer continuous monitoring of inflammatory markers directly from our body. This platform is wearable based on flexible technology and materials, and also it's ultra-sensitive because of its miniaturized size, allowing us to really detect very small quantities of inflammatory markers and detect really the disease at very early stages. It's also non-invasive, like in the form of a sensing bio-tattoo. So this actually platform, this novel platform will be a very pivotal tool for the medical practice because it will be at the side of the doctor, especially during surgeries when also organ transplants are involved, because they can signalize directly if an organ transplant is rejected, so the doctor can immediately take action. Also, it can be a beneficial tool for our everyday life, since it can monitor from our arthritis, that usually we need very harsh and dangerous drugs, till our food intolerances, and also signalize the response of a specific treatment that we are taking. And overall, I believe that this novel platform will make the inflammation, and especially hidden inflammation, from a secret killer to a powerful weapon that will promote our health and well-being, help us, and informing us about our health status and condition. Thank you very much.
Are there any questions for Julia and her great work? If not, Julia, thank you so much for making sure you actually take care of our well-being. And congrats for your work. Next is Elisa. Elisa Narminio is going to tell us how she's breaking the walls of child trafficking.
Thank you. Isn't it nice to start your day with a warm cup of tea or coffee? How many of you had one this morning? Well, that makes a lot of us potentially responsible for child trafficking then. 17 top producing countries use child labor in their plantations. 1 million children are trafficked around the world. Even worse, these numbers keep increasing, despite the fact that many good people, government officials, charity workers, police officers, put a lot of effort into combating this practice. So what are we doing wrong? How do we make sure that we can effectively protect children from being trafficked? When I was 19, I was a nanny in France for 2 months. On occasions, I would see a young lady cleaning the apartment. Some days, I would come home and find one of the apartment rooms locked from the outside. It was just months after I had left the family, through a random conversation with a neighbor, that I discovered that the young lady had been locked up in there. I had been blind for it, but people who knew had done nothing. Most of us believe that child trafficking happens in faraway countries and is caused by ruthless criminals. But it also happens just around the corner, in the home of your lovely neighbor. My job is to help identify trafficking situations. This is Chan. I met Chan in Thailand last year when I was investigating forced labor on fishing vessels. And this is Aom, his little sister. Aom is 5 years old. And this little girl of 5, has been identified as a sex trafficking victim and placed in a shelter just after my visit. Against all odds, what I discovered was that children like Aum, who are taken into care by public authorities, often end up being more vulnerable to trafficking than before. And this is true in Thailand, but it is also true in most of our European countries. My work is to break the vicious circle of vulnerability that these children are in by identifying mechanisms in our social fabric, in our laws, even in our language, that prevent us from durably lifting children out of trafficking. I develop solutions that policymakers can implement so that next time we find a locked door, we can break the wall and help a child escape. Thank you.
Are there any questions for Elisa? If not, thank you for your work on a very important topic that might be closer to us than any of us are very much aware of.
Thank you very much.
Following Elisa is Jonathan. Jonathan Kinson is going to tell us about breaking the walls of energy.
Yes. Hi, everyone. So today I'll be indeed talking about two things, energy and my obsession with the letter P. And I will start with P like precious metals. Has anyone heard about precious metals before? I can see a few hands. I can see a few rings, actually. Maybe it's wedding rings. Maybe it's gold. Maybe it's platinum. Platinum and gold, they are precious metals. And if you're single or you don't like rings, don't worry. You actually use precious metals every day. In your car, there is a tiny amount of precious metal that's used to make the car less polluting. But this tiny amount accounts for a lot of money because precious metals are non-renewable resources. So the challenge is to use it as efficiently as possible. In the case of cars, some of you will say, well, soon we will have clean cars, no need to treat the exhaust or anything, there will be no fuels involved. And hopefully that means that we will save a lot of precious metals to make many more rings. That's the ideal case. Actually, this is not what's going to happen. We're going to probably get to clean cars, but at the expense of using even more precious metals. Not to treat the exhaust anymore, but actually to power the vehicle. And I could say the same. We're going to need more precious metals for many applications in chemistry, in energy, in medicine, and so on. On top of that, you can add a growing population, the need for clean power, and ideally producing all that at a fair price. So what I did is I developed a surfactant-free colloidal synthesis of precious metal nanoparticles. So don't get scared, I'm going to explain. The first thing, what that means is the first thing is that we can control and optimize the use of precious metals down to the atomic scale. You can't do better than that. The second thing is more of a personal kind of lucky story. I don't drink alcohol, and I've been crazy or curious enough to try tequila and vodka to make nanoparticles, and it actually worked. And from there, we actually figured out that we can use very simple alcohol to synthesize precious metal nanoparticles. And it has many benefits. The first thing is that compared to other syntheses, we can produce a nanoparticle very easily in very simple solvent, and typically non-toxic solvent, as opposed to the state of the art. Second thing is that we can process the nanoparticles more easily, which means that it's easier to implement the nanoparticles in real technologies. And the last thing is that we've got more performant nanoparticles, which means up to 10 times more efficient use of the precious metal. So we quickly make sure that this was scalable and compatible with industry. This is why this technology is patented and trademarked. And at this stage, I hope you're all kind of more convinced of the power of the letter P. And more seriously, if you're driving a clean but affordable car very soon, hopefully that will be thanks to our technology. Thanks a lot.
Thank you very much, Jonathan. I see a question over there.
So it's a competing technology. Batteries is a different way of thinking of the next generation of cars. This is more for fuel cells vehicles, but actually, if you think of chemistry, that's applicable in many, many areas of chemistry, but not directly as such in batteries. But the fuel cells vehicle is a competitor of batteries and probably have many more benefits than batteries.
Thank you, Jonathan. Are there any other questions? If not, thank you for your
Thanks.
presentation and congrats. Now we have on the stage with us David Phage, who is going to talk to us about plastic pollution.
Thank you. Of all the waste we generate, plastic bags are perhaps the greatest symbol of our throwaway society. They are used, then forgotten, and they leave a terrible legacy. I like this quote from Zach Goldsmith because this exactly reflects the current plastic pollution issue. As we are so reliant on plastics, such as disposable shopping bags or food containers, we are seeing growing amounts of plastic waste going nowhere. How can we overcome this issue? Recycling and reuse have been proposed to be important solutions, but those are not enough. My solution is to replace plastics. There are some resources on the Earth that are underutilized or, in most cases, treated as waste, such as crop residues from agriculture or prawn shells from food. These bioresources are renewable and contains the so-called biopolymers, which can be used to make biodegradable materials without any harm to the environment. But it is extremely challenging to process biopolymers using conventional plastic engineering techniques. Some biopolymers are actually components in food, and humans have developed well-mature, cost-effective techniques to process food. Think about flour for making pasta. Do you know why flour becomes hard when we add water into it? Because water makes interactions take place within flour that produce forces that hold flour particles together. So, inspired by this, our research has led to a facile method to produce a new bioplastic based on prawn shells and silk. We found these bioplastics as mechanically strong as engineering plastics, which is exciting, and we've proved it is possible to make such biocomposite materials with varied recipes based on different biopolymers and other renewable additives, and also to allow powerful interactions within the material, just like in pasta. In this way, we could tailor the material performance for certain applications, from packaging to biomedical treatment, to replace traditional plastics. But our research will only make the change with all your support by adopting biodegradable and renewable materials in our daily life. And then we will make a great step forward to a sustainable and healthier future. Thank you very much.
Thank you very much, David, especially for a very strong call to action. Please, there's a question over there.
Getting boring, it's always been. Have you anything in the market yet?
Yes, there has been a lot of in the market, but the problem is that they cannot do everything. The property has to be tailored for certain applications, and this is what we are doing. We are doing composites, and we are developing formulations, and that can be easily processed, and that's the work we are doing now.
Thank you, David. Thank you very much, and we can never, ever get bored of your questions, so continue. Thank you.
Okay, thank you.
Next up is Katarzyna Sitnik. She's going to talk to us about herpes virus, actually, and the herpes virus safe house. Please.
There is a deadly virus in this room. And most of you are infected. But relax, we are safe, unless at some point in your life you might need to undergo a transplantation. Here is why. Herpes viruses, which I'm talking about, persist in healthy individuals in a harmless dormant state. However, in transplant recipients that need a lifelong immunosuppressive treatment, this dormant virus can reactivate, causing fatal disease. Despite the number of transplant patients grows by 200,000 every year, there is no available treatment that can prevent herpes virus reactivation in that group of patients. And this is the wall I'm trying to break. So why aren't there any treatments? I realized that the basic problem is the fact that scientists do not have access to dormant herpes virus for their studies. Imagine that one cell of your body is the size of my fist. Imagine these cells are arranged in a single string. You will encounter a single dormant herpes virus only once every 700 meters. Such low frequency is not sufficient to perform basic research on herpes virus reactivation. Through my research, I found a way to isolate cell populations that are significantly enriched for dormant herpes virus, meaning that this impossible distance of 700 meters is now reduced to 7 meters, finally putting dormant herpes virus within our sight and within the reach of basic science. I'm proud to say that we have made the first critical step towards protecting transplant patients from potentially fatal disease, the danger that comes from within. Thank you.
Thank you very much, Katarzyna. Are there any questions for Katarzyna and her very important research? If not, thank you so much and congratulations for the success you've just reached. And now you all know what's coming. The last presenter, the last but definitely not the least. I'm not at all increasing his stress. He's not at all looking forward to coming on the stage, but I know he's ready. So Rodrigo Fernandez Jimenez is going to talk to us about him breaking the walls of childhood obesity.
Thank you. So, heart attack is the #1 killer globally. As a cardiologist, I see a number of patients every week suffering heart attack. So these patients can develop huge complications. One of the problems linked to heart attack is obesity, which is one of the major risk factors for developing a heart attack. And obesity roots in childhood. According to worrying estimates, 1 in 3 11-year-olds in Europe is overweight or obese. The main reason lies in unhealthy habits, like the consumption of food high in fat and sugar, and low nutritional value combined with little physical exercise. So most of these children who are overweight or obese will be so as young adults. And these individuals will be at high risk of developing obesity-related diseases, not only heart attack, but also other highly prevalent diseases like depression and cancer. So how can we stop the obesity epidemic? Well, the solution is in front of us, and it is much more simple than we think. Our behavior as adults is very difficult to change. However, because the unique plasticity of the human brain during childhood, this period represents a window of opportunity to instill long-lasting healthy habits. At my institution, we have developed a comprehensive and innovative school curriculum to teach the children healthy lifestyles. So we teach them how the body and heart works, a healthy diet, how to keep a physically active life, and the management of emotions, which is very important at this in the children. We train the teachers, who are the ones who actually deliver the intervention. And we also do activities with the families, and we give recommendations for the school environment. Using this strategy, we have successfully delivered this educational program to more than 50,000 children at different educational stages across 3 different countries. So the solution to break the wall of childhood obesity is here, and it's simple. Rather than teaching children to memorize useless things like the sign of pi, we should teach them how to live a healthy and sustainable lifestyle. Thank you.
Thank you very much, Rodrigo. Are there any questions for Rodrigo and his research? If not, thank you so much for actually closing the series of our presentations with a very strong performance.
Thank you.
I would really kindly ask you to give a very, very big round of applause to our very brave researchers here today. This has been a very important step, I would say, both in their lives as well as in their careers, because we've just put them on the spot massively. So thank you so much for actually being up to this challenge. Now, the next part comes, and that is I would kindly invite the jury to leave the room and to start discussing about shortlisting the three top finalists, out of which the winner will go to the Falling Walls Lab final in Berlin on the 8th of November. Thank you so much. In the meantime, we will all stay here, and they won't know what is it that we're doing or we're talking about. Thank you, thank you. To the ones that actually need to leave, I would kindly ask you not to forget to give us your ballots, your votes for the preferred candidate or the preferred scientist. I would now like to invite here on stage Bodo Richter, the Commission's own Bono. I've seen him speak, and I can tell you he does deliver exactly like Bono does. Now I raise the bar. Now he'll need to sing. Bodo is going to address you a few words. He's the Deputy Head of Unit for Marie Skłodowska-Curie actions. Bodo, you have the floor.
Yes, please. Thanks a lot. Yes, it was an incredible session. I think we've seen a very large number of very good presentations. So again, congratulations for what you did. You're all financed and supported by the Marie Skłodowska-Curie actions. It's very hard to stand here, I have to say, because now I feel it. It's actually much better to be here. But anyway, I will stay here. But it's a tougher job than I thought, actually, to stand just physically. Yeah, just physically. Yes. The Marie Skłodowska-Curie actions, yeah. So you all went through selection procedure. You all were selected, and you're now mobile. You're doing your research. You're training abroad, and I see that you're doing a fantastic job in finding solutions to problems we face every day. We face on a global scale. My task now is to fill a little bit the time between, now in 4:20. I think we will start with just handing out participation certificates to all the fellows who performed here today. You all, as I said, were selected. You've heard it in the beginning. There was a lot of interest in coming here to Brussels to do this. You are the 30 lucky ones, and later on we will have the one who will go to Berlin in November. Yeah? But before that, I think we can just move on and give you the certificates. And after that, I would say we can have a little discussion about the Marie Skłodowska-Curie actions. You could probably tell me what you like about it, what you don't like about it, give me some ideas for the future. Or if you have any questions about program implementation, we can also talk about this. And by that time, I think the jury will come back and pronounce the winners. Okay, shall we?
If I could add, I think we should raise a challenge for Bodo. You know, because we've all been paying attention to your presentations, you should quiz him on it.
On the presentations?
On the presentations, exactly. See whether he's been paying attention, just saying.
Well, I think I paid quite a lot of attention, yes. And, but to be honest, when you get these 30 presentations, one after the other, it's very difficult to remember what you heard at the beginning. Yeah? It's very difficult. And so I do not envy the jury, which now has, I mean, of course they took notes and everything, but who has to take a decision, a common decision on the 3 strongest performances. And by the way, whom am I supposed to give this? Yeah, have you all handed in your voting sheet? Oh, okay, I'm no longer interesting for the young ladies apparently. Okay, good. That's okay. Yeah.
You can call out the names if you'd like, and then I will just invite you to come one by one here in front,
Yes.
and we'll also take some pictures.
Okay. So I can.
Brilliant. Thank you so much. We'll start from the beginning. Lieselot de Wachter. And congratulations.
Okay, good.
We need to make the pictures perfect as well, of course.
I feel like a rock star.
Nice. Yeah. You are. Congratulations. Next up, Bruno Rodinho Janeiro. Bruno? Bruno. Thank you.
Also, you feel so famous, yes?
Next up, Tinkara Tinta, also known as the jellyfish dancer.
Very energetic performance, I have to say. Yes, here you go. Congratulations. Thank you.
Thank you. Next up, Francesca Forno. It's much stronger than me. You killed me, Fulvio. You killed me. Congratulations, Francesca. It also shows I need glasses. Fulvio Flamini, please. And congratulations.
That's what I call style.
This one, I will get this right. Ariadne Jebala Kukula.
Well done, Ariadne. Thanks for taking part in this. Thank you. Sorry.
Next up, Harita Corliel. Harita, please.
That's also Sharon.
And actually, the correct way should be Benjamin Raymond. Is that correct? Benjamin Raymond. Thank you, thank you. I need to work on it.
Oh, Australia! Wow. It sounds. An Australian with a very Quebecois name, I find. But anyway. Thank you.
We should actually do the test. You know, who has traveled furthest? Have a special award for that as well. Thomas Winkler. Thomas, congratulations. Thank you. Next up, Rita Branco. Did you check the water we have here?
Congratulations. And anyway, it's too late. We drank it already.
Thank you so much.
Benjamin Droguet. Is that also correct? Yeah.
Okay, that's French. French. Okay, Benjamin. Félicitations.
Now we're going to Greece. Athanasios Rogdaikis.
Wow. I think you did a good accent.
I've been there.
Congratulations. Okay, here we go.
Nicolas Robinson Garcia. Congratulations, Nicolas. Thank you. Next up, we're going to Argentina. Mariana Perez Ibarreche.
In Ireland, okay.
Thank you. Thank you very much.
Cristina Macondo. And we also have your resources over here. You should circulate this. This is incredible. I would definitely fall on this versus this.
You just, it's also a huge difference in weight. Yes, yes, yes.
Now we're doing the testing period.
Thank you.
Please. Ivana Cavaliere. He knows he's up. Josie Esteban Rodriguez Condia.
That's a mouthful, no? small.
Ivan Palaya. Mafalda Ferreira. Thank you.
I know I'm Portuguese, but I'm in Amsterdam.
Ah, in Amsterdam. Yeah. Okay, congratulations again. Yeah.
Gillian Bracht.
Nederland. Yes.
Next up, Mercedes Regadio. Emmanuel Salifu. We need to make Emmanuel now to calculate how much soil has been eroded since the beginning of his speech. See whether he's been calculating.
And where is, by the way, this huge fungus? Where can we find this one? It's in the US. In the US? Oh. Okay, good.
Now he tells
Everything
us.
is in the US anyway.
Now he tells us. Wojciech Rzadkowski. Julia Tsuwadajki. Elisa Narminio. With possibly the youngest scientist out there. She's been listening to all of your speeches, by the way.
Thanks a lot, yeah. I knew there were lots of Erasmus babies, but apparently there are also lots of Marie Skłodowska-Curie babies nowadays.
You should put it in your new stats. You
Yes,
need to do that.
definitely.
So I think it's Jonathan Kinson, am I right?
Ah,
Jonathan Kinson.
Friend?
Thank you. David Fengwei Kshe. Katarzyna Sitnik. The person telling us we all have this virus, thank you. Rodrigo Fernandez Jimenez. Congratulations. Thank you. I actually have a challenge. Would it be possible for us all to get on the stage so that we can take a group photo?
I guess
Yes?
so, no?
We do both a group photo as well as a very popular selfie.
Yeah, so please, just come here. We do group, yes, and the selfie. But who has the longest arm to take a selfie?
We'll test. We'll need to measure. We have a lot of scientists in the house. Who can quickly say? Very good. All the families of everybody present here will now see how quickly is it that we all gather for a selfie.
Thank you. You are not one. Usually I just eat meals. I think I can go back and eat meals.
Put it on.
Yes.
So this is class of 2019. I think this is the line from Star Wars.
Thank you.
Wait, I'm doing this.
Can you also please do it with my phone, otherwise?
Yes, and one a little bit more funny, like that. Let's go, everybody. Yes, thank you.
Nice, congratulations, thank you.
Okay, so don't fall.
Yeah, I was going to tell you.
I will give the microphone back to Bodo so that he can tell us a little bit about Marie Skłodowska-Curie actions and how is it that you're supporting researchers and what are the criteria to support the researchers?
Okay. So let's first take a seat. Okay, I'm not sure you're all very familiar with the structure of the program and the program itself. So the Marie Skłodowska-Curie Actions, it's a program that has been running for more than 22 years. Yeah, I think this year we have reached the cap of 130,000 researchers we have supported in their mobility, in their training. But of course we do this not only because we believe in bright people and we want to help you in your future, but we also think that what you do and what then the result of your research will be will contribute to a more prosperous society and a more competitive economy, new products, new services, and so on. In the end, I mean, I don't know if you've had the time to also go to other sessions. I don't think you had the time actually, but there were other sessions running today where people said, why do we need fundamental sciences, for example? Why do we need this research? Yeah? And the Marie Skłodowska-Curie Actions are one of the few programs like the ERC that support research in every kind of area, at every stage of the research. Some good arguments were, because you never know what this is good for. Yeah? If we hadn't done some research 50 years ago about, now I don't even know anymore about it was, we wouldn't have a mobile phone now. But it was that research 50 years ago that allowed us to create mobile phones, and now we cannot imagine a life without a mobile phone anymore. Yeah? So, this program is quite successful. Yeah? But that's also the problem of the programs, because, I mean, I don't know if you know, but for these innovative training networks, for those PhD students among you that participate in an innovative training network financed by the Marie Skłodowska-Curie Actions, the success rate of these networks is just 7%. It's very, very low. So you are participating in the top-notch doctoral schools or doctoral training programs at European level. Yeah? And for the individual fellows among you, there the success rate is between 13 and 14%. It's also very low. So also here you can be proud of being selected by the program and to be financed by the program. So we have, in essence, we have 4 actions. So we have these doctoral training networks, in innovative training networks. We have individual fellowships. We have RISE. I don't know if you've heard about RISE, Research Innovative Staff Exchanges. That's the exchange program to help organizations, research organizations, but also universities and other academic players to collaborate with non-academic sector. So to make this knowledge transfer happen more easily in Europe. Yeah? Then we also have CoFund, and also I think here today some of the researchers that are here participate in a co-funded program, for those who don't know what it is. So we give some money to national, regional, or even institutional programs to make them follow more or less the rules and the best practices of the Marie Skłodowska-Curie reactions, notably in terms of selection of the researchers, of the freedom you get to do your research, of your working conditions, and so on and so on. Yeah? And then, of course, we have the European Researchers' Night, which is a small action, but it reaches out to a lot of people in Europe on every Friday at the end of the year, at the end of September, sorry, so tomorrow. Yeah? It's the European Researchers' Night. Probably some of you will participate in it. If not, please consider at least going to visit one. There are 370, it takes place in 370 cities around Europe tomorrow. Maybe there's one just close to where you're from or where you go to tomorrow. And some of these events also take place actually on Saturday. Yeah? Now we're at the more or less a turning point from Horizon 2020, the framework program for research innovation, which lasted until 2020, so one more year to go. And from 2021 on, we will have another research framework program which is called Horizon Europe. For that, we have decided not to change too much when it comes to the Marie Skłodowska-Curie reactions, because I think it's a program that works. And I hope you all agree that it works. However, I would be very much, so we will continue having these 5 kind of actions. We will give them a little bit different names, because for outsiders, for example, it's not always very understandable what an ITN, Innovative Training Network, is. Yeah? So maybe we will call these ones just MSCA doctorates, because they're all about doctoral candidates and doctoral studies. Yeah? And the individual fellowships most probably will become MSCA postdocs, yeah? Because then you know to whom this is addressed to. Yeah? RISE is a little bit long, research and innovation staff exchanges. We'll just shorten it a little bit and make staff exchanges out of it. And co-fund will remain co-fund. Yeah? And about the European Researchers' Night there, we are still thinking, but one idea is to call it the MSCA and citizens, because it's going out to citizens. It's a kind of outreach activity. We would like to have many more fellows and many more organizations in Europe to step out of their labs and talk to the general public, to explain to them why they do their research, what the research is good for, and as I told you already yesterday, and also to make them, or to reassure them that the money they pay in form of taxes is well spent. Yeah? And through the training you had today, yesterday and today, I think you can feel much more confident now to go out and also do this job. Yeah? And by the way, every fellow which is funded by the Marie Skłodowska-Curie Actions has the obligation to do some of these outreach activities. Yeah? So I think now you can go out much stronger than you were before you came into this training. But is there anything, and I'm really interested to hear from you, is there anything where you think this doesn't really work, or it could be improved a little bit in the Marie Skłodowska-Curie Actions, in your own fellowship, something where you think, yeah, if I only had known this, or if I only had a little bit more money here or there, I could have done this. Yeah? So is there anything where you think we can improve, or something that you find disturbing during your own fellowship right now, during your research? You have some issues, yes, yes, finally. 2 ladies have issues.
I just have, because we are doing a PhD in basic research, is really challenging to do it in 3 years. So I think that's one of the main challenges. Of course, this includes more funding, but 3 years is a really short time to do secondments and to do and to do a PhD, to finish a PhD. So maybe 4 years would be a better option, I guess. especially because of the secondments that we do. They are amazing, of course, and they are really productive. But then it's maybe half a year abroad, and then the rest of the time is a really short time, I guess.
I have one small thing to add to it. I was in a course of grant writing, and there they explained that for the individual fellowship, you need 4 years of experience or a PhD. No, 3 years of experience or a PhD, I think. No, I'm confused. 4 years. So if your PhD is 3 years, you need to bridge that 1 year to wait to apply for an individual fellowship, for example.
Well, the rule currently is either 4 years of experience minimum for an IF or a PhD degree. So if you have a PhD degree, of course, you can apply at the time of the call deadline. Yeah. So normally, if you can get your PhD within 3 years and you have the title of the PhD, it doesn't matter. These 4 years do not matter anymore.
No, well, there's still sort of a bridge
Mm-hmm.
because the application and actually getting the results
Yeah.
usually is a little bit longer. So when you have your PhD, you can apply, but then still you have to wait until the outcome is there.
Yes. Yeah, but somewhere we need to. Yeah. Yeah. Okay. I mean, about the duration of the support. Yes, I know. We support innovative training networks, the projects themselves, for 4 years, but then the individual PhD candidates only for 3. Yeah. I mean, this is a little bit in line with the Bologna process, where we have the 3 levels of high education: bachelor, master, and PhD. And for the PhD, there should be a rule that they should last 3 years. Yeah. However, of course, I know in some countries, in some areas, they last for 4 years, and 3 years is really tough. I also admit this. Yeah. But as I said in the beginning, our innovative training networks have a success rate of 7%. Yeah, 7%, 8% sometimes. Huh? So if we now would say each fellow will get 4 years in the project, first of all, this would mean each project would get 25% more, or 33% more, because we get 3 years with 33% more funding, yeah, and which would reduce the success rate again for these training networks. Yeah? And of course, it would reduce the number of PhD candidates we can finance. Yeah? At the same time, we also tell the organizations that are involved in innovative training networks that they should make sure that there's also, if your PhD takes 4 years, that make sure that you have some follow-up financing for the 4th year. Yeah? Okay, I mean, the best thing I think you could do is go out there and scream very loud for more money for the Marie Skłodowska-Curie actions. Yeah? Because for the moment, I mean, for the period 2024 to 2020, we have a budget of 6.2 billion euros. Yeah? For the next 7 years, for the moment, the budget is proposed to be at 6.8 billion euros. So it's not a huge increase. Yeah? If you want to have a little bit more flexibility in all these kind of things, and us to be able to finance more researchers and to finance them for a longer period, we need more money. If we can't get more money, we need to cut somewhere, and this is one of the ways we cut. I mean, I also hear a lot of times that individual fellows, these 2 years, are very tough. Yes, yeah, but there it's the same problem. It's the same problem. One thing we changed recently is, for example, that for reintegration grants, I know if you, or for career restart grants, for career restart grants, we said for those people who, let's say, for maternity reasons, for example, or for parental leave, stopped for research for a certain time, it's a little bit more difficult to get back into research and then to have a full fellowship and do all the deliverables and all the things we asked for in 2 years is a little bit tough. For that group, for example, we now extended the fellowship up to 3 years. But of course, this has an impact again in terms of the numbers of people we can finance. Yeah. But there were other questions here.
No,
yeah?
just maybe.
Yeah.
In addition to what was phrased before, the same applies when the same applies when someone is called to fill an open spot when someone resigns from the program. So the funding is for a shorter period of time, yet the amount of the project that you have to do is as if you're starting from the beginning. So, like, yeah, I just wanted to put it up there.
Okay.
Yeah, my question or remark is not anymore from ITN, it's for individual fellowships. So when you get a grant, compulsory you have to change and move to another place. In some cases, you are going to start working with a team that you don't know from before. You just know them because of one Skype call or a research paper, and you interact, and they say, okay, yeah, if you want to try it, yeah, let's try it. So in principle, in the proposal, there is the supervisory team and the researcher. Then the researcher do the work, and how I was thinking how to ensure that the supervisory team is engaged with a project when they don't feel it like compulsory to be engaged. And I don't know if I explained correctly. Is there a way just to scare them, like like sending an email, like, you know, we are going to evaluate how is the performance of the supervisory team. So not only like to, yeah, like just to be sure that they don't feel that they don't have any problem if they don't do what they should be doing.
Well, I think the supervision of the individual fellow should have been explained somehow.
Or the support. I mean, what is the stay in the grant agreement, but like...
Well, I mean, I guess normally you should have received a copy of the grant agreement. Yes. Okay, so that's already good. Then you see what all the obligations that are in this grant agreement for the beneficiary and for the supervisor. The beneficiary being the host organization where you at. Yeah. I would assume that in the proposal there was, for example, there was written something about how they're going to supervise you. Yeah. So if you as a researcher feel that there is a lack of supervision or a lack of interest in what you're doing, I think first of all, of course, we always need to talk to the people that are supposed to be doing this. But if the problem is persisting and you see there's no take-up of, you know, of the task that you would like them to do, I think it's always good to get in contact also with your project officer at the research executive agency. Yeah? I mean, I hope you all know who your project officers are at the research executive agency, at least individual fellows, no? Yeah? Because they may be able to help you. Yeah? But I think I would take this as a very last resort, because normally, I mean, it's always better to speak between people and to not to scare them through. The commission will come and do an audit, and you know, you may have to pay back all the money you received. No, no. But I think, I mean, there is something in your proposal that says you should receive supervision, you should receive it in a certain way, you should get a career development plan. This should be revised probably after a couple of, after 1 year, for example, to see how you progress. So this is the way, I mean, you know what's in the application, because I think you helped write this application, or most probably you wrote it yourself, yeah? Yeah. So this is not just a one-way street that you go somewhere, do research, and you're all on your own. It's always also, the project has been evaluated, you in collaboration with the host organization, and what they promise in the application, they should also deliver. And whenever there is a problem in delivering things, but not only in terms of interaction between you and the host institute, but also in terms of your own deliverables, yeah? As soon as you realize I'm a little bit behind or have to change a little bit, yeah, go and talk to your project officer and the research executive agency, because it's not fair to that person then who normally follows your project that after your project has finished, all of a sudden you submit a report and there's written, I did something completely different. Yeah? So they should always be aware, and then normally there's no problem that you change things. Yeah? Because life is full of changes, of surprises, also research projects. Yes?
I have two questions actually. One is very specific concerning ITNs. One is very specific and it concerns secondments. So this period abroad that it's compulsory to do. Sometimes these secondments were decided 5 years ago when the group was writing the proposals, and sometimes they don't make sense at the moment, at the time, and at the place where you're doing your research. And I got the feeling when I met our project officer that it's actually very strict, the rule to. There's a strict rule concerning these secondments, and you have to do them. And I think it might be made less strict because sometimes it's really. Yeah. And the second question concerns the evaluation process. So maybe it's a more general question. And in our network, there's this sort of joke going around that concerns evaluation because our project were presented, I don't remember, 3 or 4 times. Well, the first 2 times it was rejected with some suggestions, modifications where they did some modifications. The third time it was presented with new modifications, it was rejected. And the fourth time, without, and he also had a pretty low score, the fourth time it was represented just for fun, without any modifications. The same project that they had presented the year before, and it got the top score. So,
yeah. Okay, your first question, secondments. I mean, all depends also in ITN. Now it become a bit technical, but there are 3 different modes of ITN. There's European training networks, like normal ITNs, but then you have industrial doctorates, you have joint doctorates. Yeah, and of course, for some industrial doctorates, for example, you need to spend 50% of your time in a non-academic partner. Yeah, so there, this collaboration is very, you can't change it, these secondments. Others, I think if you start talking to your project officer in, or your host organization, yeah, at REA, about the meaningfulness of secondment for that particular person in the ITN, I think there are things to be reconsidered. Normally, I mean, we're not just like this and don't see what's going wrong. I mean, things change over 5 years' time. Yeah? So I would try it again. And then the other thing is evaluation procedure. I mean, the thing is, the evaluation is always completely independent. Yeah, and which means we get experts from the research world across the entire globe, and 1 year, 3 people or 4 people, researchers, look at it. Next year it's 3 others or 4 others, and they compare this also to the others that have been submitted. So every year is a different game. Yeah, and sometimes, yes, I'm sometimes surprised that in 1 year you get 89%, the following year all of a sudden it's 60, and then whoop, it goes up to 92. But this is because independent people look at these things, often different ones. Of course they talk to each other, and at the end, if it's a complete, I mean, it's a resubmission, they are told this project was submitted last year. Yeah, but often they do not change anything in their evaluation because they say, no, we look at this completely independently from what has been submitted before. And this is the reason why you sometimes get these evaluations where you think this is not possible. Yeah? But I think they all do it to the best of their knowledge. And sometimes, yeah, I mean, we try to make it as fair and as objective as possible, but we are all human in the end. And when you get ITNs, for example, it's 1,600 applications per year, 130 are financed. Yeah? So there is a lot to be processed, and it's even worse for individual fellowships, 10,000, and then 1,500 are being financed. Yeah? So I'm not excluding that there might be once in a while a small mistake somewhere. Yeah? But normally, the marks you get represent your position in that year's evaluation exercise. That's it.
Thank you. And I think we have one last question from Emmanuel.
Okay.
I'll be quick. Okay, I first of all it goes without saying that the Marie Curie PhD is probably the best anyone could ever have anywhere. So,
okay, quoting on Twitter.
Yeah.
Thank you.
And as an international student coming from Nigeria, I consider it like a privilege to actually have the opportunity. But the challenge I had all through my 3 years was with the mobility was great, but the fact that I need visas to move. Every 6 months, I had to apply for visas to go for different network meetings and conferences and all of that. And I think some individual fellows face the same thing when they leave the European Union to other places. So if there's a way for future participants of having maybe like a welcome kit that makes the project officers or coordinators know about the special considerations for such international students, so that if it means giving, for instance, I was recruited in the UK and then I had to work also in Europe, and I need a Schengen visa every 6 months. If it's possible to get both visas for 3 years for the whole period, so it's all sorted from the beginning, it would probably make more sense.
Yeah. I mean, the visa issue is tricky because it's not really the responsibility of the EU to issue visas. Yeah, but we have visa directives, and which means laws that should be implemented into national law. And there is a research visa directive and there especially mentioned are MSCA students and fellows, PhD candidates and fellows. But of course, this takes time to be implemented, and then you should get a visa that covers your research period, your training in Europe. But it is something where we have very little influence on. Yeah, normally your host institution should give you all the help possible to obtain the visa. Yeah,
I had all the help actually. So, but my thought is if there was maybe like a letter from the commission or something at the beginning to say this person is recruited under this program, and you can give them a Schengen visit visa for 2 years or something like that, or 3 years.
I think this is
probably help more.
I mean, we're not there yet.
Okay.
Yeah, we're not there yet. But I think once this directive is actually put into national law, then it will become much easier.
Thanks.